Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will learn the concept of stereochemistry of amino acids. Okay, let's get started. Now coming to the stereochemistry, this we have already discussed in the previous class, haven't we? Okay, let's recall it quickly. What is stereochemistry? It is the study of how atoms are arranged in a 3D space, right? Now in amino acids, how the stereochemistry is important? Because here the proteins and enzymes, they are shape specific, right? Means they interact correctly with the molecules that have the right 3D structure. Okay, now let's see in detail. So next topic is chirality in amino acids, right? So here we have already discussed about this chirality, right? What is actually a chiral carbon? A carbon when it is bonded to 4 different groups, right? Now if you take example of an amino acid, right? So here carboxyl group, hydrogen atom, amino group. Now this is the R group which differs, right? Now here these are all again different groups. So here when the carbon atom is bonded to 4 different functional groups, then the carbon becomes chiral. So here it is the first carbon, alpha carbon, right? Now this is nothing but the chirality. Now let's take an example of a glycine, right? If you take example of glycine, amino acid, so this is the general structure of amino acid, only the R group differs. Now in glycine, what is the R side chain? It is a H. Now is it a chiral? No, because here if you notice, glycine contains 2 same functional groups, so it is achiral. Is it clear? Now let's go to the topic L&D configuration. Now this configuration comes from the Fischer projection. This we have already discussed, right? Fischer projection means open chain structure. Now L&D configuration comes from the Fischer projection and is based on glyceraldehyde example, right? This we have seen in the carbohydrates. Now here, how to differentiate L&D configuration? If the amino group is on the left side, then it is L amino acid. If the amino group is on the right side, then it is the amino acid. Let's take an example of alanine. So for alanine, R group is methyl group, right? So actually this is a general formula. So for R, it is methyl group. Now it is the structure of alanine. If the amino group is on the left side, then it is L-alanine. On the other side, if the amino group is on the right side, then it is D-alanine. Is it okay? So here, this is how we have to differentiate L-alanine and D-alanine based on the amino group configuration, whether it is on the left or on the right. So here in nature, if you see, all the proteins are made up of mostly L amino acids only, right? All the proteins are made up of L amino acids only, but you can see D amino acids also. In rare cases, they are mostly found in bacterial cell walls and antibiotics. Is it clear? Now let's see R&S configuration. Now this R&S configuration comes from CAN, Ingold, Prelog, means priority rules. Means this, in short, we call it as CIP rules. According to this, we have to follow some rules. Now I'll explain this with example alanine. Pay attention. So here, if you see the alanine structure, let's see. First, we have to assign the atoms which are attached to the alpha carbon or chiral carbon, that too based on the atomic numbers. So here, first amino group gets first carboxyl, second methyl, third, and least hydrogen atom, right? Now we have to trace the sequence 1, 2, 3 towards right. The orientation is towards right, right? So it is clockwise. It is clockwise. Orientation is towards right, means it is rectus. So this is our alanine. Now let's see how S-alanine differs. Carboxyl group, amino group, methyl, hydrogen. Again, we have to assign atoms which are attached to the alpha carbon based on the atomic number. Amino 1, carboxyl 2, methyl 3, hydrogen 4. So, if the see again trace the sequence 1, 2, 3. If the orientation is towards left, counterclockwise, means sinister, means left. So it is S alanine. Is it clear? R alanine and S alanine according to CIP rules. So okay. So mostly here the L amino acids are especially S configuration only, except cysteine. Let's see. Carboxyl, amino, hydrogen. So this will get higher 2 and 3. 1, 2, 3. So it is towards right, means. So this is R configuration, R cysteine. Is it clear? Why it is R configuration? Before we are saying that all L amino acids are S configuration only except cysteine, because due to the sulfur high atomic number, now this cysteine configuration has been changed to R configuration, that is R cysteine. Is it clear? Now let's see the next topic, that is optical activity. What is an optical activity? This will tells us that the chiral molecule can rotate the plane polarized right or not. If it rotates towards right, then it is dextrorotatory. And we have to assign symbol as plus. If it rotates towards left, means levorotatory. Minus. Again, don't confuse with D by L configuration and this plus and minus. Both are different. These both are different, okay? Now coming to special cases. First, glycine, it is achiral and optically inactive. Let's see. Achiral means carbon contains, actually chiral means carbon contains 4 different functional groups, right? So here it contains 2 same functional groups. Here it is the structure of the glycine. So It has two same functional groups. That's why it is achiral. And it is optically inactive because it cannot rotate plane polarized light towards left or right. That's why it is optically inactive, right? So second, threonine and isoleucine. These two are the amino acids that contains two chiral centers. If two chiral centers are present, there is a possibility of more than two stereoisomers are produced. Mostly amino acids contain only one chiral center, but here exception is threonine and isoleucine contains two chiral centers. There is a possibility of four stereoisomers to be produced, right? Is it clear? First, let's see biological importance. First, proteins function only with L amino acids because they are evolved to function with that orientation only. Means they recognize only this orientation only. Second, if it is wrong structure is present or wrong stereochemistry is there, means there will be no biological function. Third, it is important in protein folding, enzyme function, and third, correct recognition. Is it clear? Now let's see the representation of amino acids. First, Fischer projection. This is a 2D structure form for easy classification. This we have already seen, right? Mostly whatever the structure I write. Example, if you take lysine, this is Fischer projection only. Okay. Second, wedge dash models. This shows the bonds coming towards or away from you. I'll explain this with example. So here wedge dash models are what? These are 3D. These are also to represent 3D structure only, but on a paper we represent with a 2D structure. Let's see. I'm taking example of alanine. If you see, these are dashed lines and this is wedge form. These are solid lines. Is it clear? This is a wedge and dash model for alanine. So here these dashed lines are the lines which are showing away from us, and this wedge form towards. Is it clear? Dashed lines are away from you. Wedge form is towards you. And finally, 3D models. These are helpful mostly in protein folding, right? So I'll give an example with a basic structure of amino acid. This is the amino group, oxygen, this is a H group, and this is the R group. Is it clear? This is the 3D model. Let's see how it connects to real life. First, we are taking taste indifference. Example, aspartame, L and D forms, L aspartame and D aspartame. Now, L-aspartame taste sweet while D-aspartame taste bitter. So there is a taste in the difference. 2. Bacterial cell wall contains, this we have already discussed in the beginning, right? D amino acids only. Why? Because they help them to resist enzymes that target L forms. Third, misfolded proteins. Misfolded proteins are formed or produced due to wrong stereochemistry or wrong structure that may lead to the disease Alzheimer's. That's all for today's video. I hope you have understood the concept. If you have any doubts regarding the concept, please comment on your doubts. Okay, and if you found this video helpful, please like, share, and subscribe to Biochemistry by Revati for more simple and clear explanations. Please do support me. And in the next video, we'll move to the second unit, that is carbohydrates, right? If you wanna check the playlist of total unit 1, I'll give. So until then, keep learning and stay curious. See you in the next class.